Welcome to BioVivacious. BioVivacious is a YouTube channel dedicated to clear fundamental concepts in biosciences. In an earlier video, when we were discussing about the characteristics of the enzyme Rubisco, we have looked at the two catalytic activities of this particular enzyme. This enzyme can function as a carboxylase as well as it can function as an oxygenase. When it is functioning as a carboxylase, it will be producing three carbon products, whereas when it is functioning as an oxygenase, it will produce one 3-carbon product and one 2-carbon product. So therefore, when it is functioning as an oxygenase, it is less productive, which also means that the biomass of a plant will be reduced. When is it acting as a carboxylase and when is it acting as an oxygenase? When the temperature is less than, let's say, 28 degrees Celsius, it will function as a carboxylase enzyme. When the temperature is greater than 28 degrees Celsius, it will function as an oxygenase enzyme. We know for a country like ours, where the temperature is generally very high, the plants will switch over to oxygenase reaction and it will end up by not growing properly or producing less of biomass. Many plants have made an adaptation in their structure in order to combat this difficulty. What these plants have done is, instead of opting for a C3 pathway, C3 because the first product which is formed is 3-phosphoglycerate. Instead of opting for a C3 pathway, it has opted for a C4 pathway, which means the first product formed will be a 4-carbon product, which is nothing but oxaloacetic acid. This video is about how a plant is carrying out the C4 pathway. When we look at the C3 pathway, we know that the carbon dioxide is taken inside by opening the stomata of the plant. So this is, if you consider, this is the stomata of a plant. This is the stomata. So all the mesophyll cells, bundle sheath cells will be up. This opens the stomata and carbon dioxide is taken inside. When carbon dioxide is taken inside by opening the stomata from the air, this is the air, there is a possibility that water will come out. In a dry climate, when a plant is trying to open the stomata in order to take up carbon dioxide, it will lose a lot of water. So this is a typical problem with the plants in the hot environment. Generally, it has got 2 guard cells which will try to close the stomata so that the water loss is minimized. A C3 plant would require somewhere around 700 to 1300 moles of water in order to fix 1 mole of CO2. This is a huge amount of water that is required by the C3 plants. Now, with this adaptation in the C4 plants, what it will do is it will concentrate carbon dioxide. By doing that, a C4 plant, this is the case of a C3, and a C4 plant is able to fix the same amount of carbon by using half of the volume of this water. That is about 400 to 600 moles of water is used in order to fix 1 mole of carbon dioxide. So this is one of the biggest advantage of C4 plants. So we are going to look at how exactly this mechanism operates. Naturally, a question will arise in our mind how the C4 plants manage in order to concentrate carbon dioxide. The C4 plants have a typical anatomy in the leaf structure. Let us look at the leaf structure in a C3 plant. So this is in a C3 plant. It has got an epidermal layer. This is also epidermis. So in the epidermal layer, then it has got mesophyll cells. Mesophyll cells, then it has got bundle sheath cells, and this is the xylem and the phloem, which is the vascular bundles. And you will find that the mesophyll cells these are the mesophyll cells. They are scattered. They are not properly arranged around the bundle sheath cells. When you look at the C4 plants, this anatomy, the leaf anatomy is very different. This is the epidermis, and this is the xylem phloem, that's the vascular bundles. Around the vascular bundle you will find the bundle sheath cells. Bundle sheath cells are fully covering it, tightly packing them. And surrounding the bundle sheath cells, there are these mesophyll cells. They form a very tightly organized packed structure. There is a direct contact between the mesophyll cells and the bundle sheath cells. Generally, there is a kind of a layer around the bundle sheath cells which is known as the suberin layer. This will have peculiar substances, waxy substances, which will prevent the loss of carbon dioxide once it is getting concentrated, or exchange of gas is not allowed. So this kind of an anatomy is known as the Kranz anatomy, which is peculiar in the C4 plants. It is because of this peculiar arrangement the C4 plants are capable of concentrating carbon dioxide. We know that in the air, the carbon dioxide concentration is about 350 parts per million. When carbon dioxide is taken inside, it becomes about 8 micromolar concentration of CO2. And at 8 micromolar concentration in a C3 plant, Rubisco will be able to fix carbon dioxide. Whereas in the case of Kranz anatomy, what it does is, here it will be about, with the less carbon dioxide, it will be about 6 micromolar. And with that 6 micromolar concentration, the carbon fixation becomes a very inefficient process. What it does is, it will concentrate carbon dioxide into the bundle sheath cells. That is to about 70 micromolar concentration. And by concentrating carbon dioxide to 70 micromolar, it is able to carry out the photosynthesis or carbon fixation in a very efficient manner. So let us look at a graph, represent this carbon fixation by using a graph. This is a simple diagram which is showing the difference in the carbon fixation in a C3 as well as in a C4 plant. If you look on the x-axis, we take the light intensity, and on the y-axis we are taking the rate of photosynthesis or the micromoles of carbon dioxide that is fixed. It's very easy to find that in the C4 plant, the carbon fixation is much higher under the given intensity of light in comparison to a C3 plant. This is one of the biggest advantage of C4 plants. In order to understand how exactly the carbon fixation occurs in the case of C4 pathways, this diagram is drawn here. This is a mesophyll cell that is segregated by a bundle sheath cell. Look at the previous figure of the Kranz anatomy. They were tightly they were adjacent, so they are separated by the plasmodesmator. This is the plasmodesmator that will act like a kind of a channel for molecules to pass through. This is a chloroplast, both are chloroplast. This is the chloroplast in the mesophyll cell, and what generally happens is a molecule of carbon dioxide enters. Now this molecule of carbon dioxide, this will be converted to HCO3 bicarbonate ion by using an enzyme carbonic anhydrase. If the first step is carbonic anhydrase interact with the phosphoenol pyruvate. So I'll write CO2 here. If the phosphoenol pyruvate in the cytoplasm, carbon dioxide interact with the phosphoenol pyruvate. PEP is a 3-carbon compound, one more carbon is added, so that is getting converted to oxaloacetic acid. And this is catalyzed by an enzyme known as PEP carboxylase. PEP carboxylase will attach a CO2 and we get oxaloacetic acid. Now once the oxaloacetic acid is formed in the cell cytoplasm, this is taken inside the chloroplast. The oxaloacetic acid will undergo a reduction reaction with the help of an NADPH. The enzyme is malate dehydrogenase. Oxaloacetic acid is getting converted to malate. Malate, once it is formed, this will be transported through the plasmodesmator into the bundle sheath cell. It is transported to the chloroplast in the bundle sheath cells. In bundle sheath cells, the malate will be converted to pyruvate. Malate is a 4-carbon compound and pyruvate is a 3-carbon compound, and the CO2 molecule is released. So this is catalyzed by an NAD-dependent malic enzyme. Now the CO2 which is released is taken up by the Calvin cycle, and triose phosphate will be synthesized. Now what happens to the pyruvate which is formed in the bundle sheath cell? This pyruvate must be transported back into the mesophyll cells in order to have the continuation of the pathway. So this pyruvate, there is a pyruvate translocase that is transported back into the mesophyll cells. Now in the chloroplast, the pyruvate will be converted to phosphoenol pyruvate. That is catalyzed by an enzyme known as pyruvate phosphate dikinase. So in the reaction, what happens is an ATP will be converted to an AMP and pyrophosphate, and pyrophosphate will be cleaved into 2 PI molecules that will drive the reaction forward, and PEP is formed. Once PEP is formed, it will continue. This is where we have began. This cycle continues. In the process, if the CO2 that has been concentrated here, this CO2 is getting incorporated into the structure and it is transferred to another location. What effectively happens in the C4 pathway is if the carbon concentration is happening in one place and fixation is happening in another place. So the C4 pathway utilizes 2 different locations in order to carry out carbon fixation. By using 2 different locations, C4 pathways becomes a more efficient carbon fixing pathway than in the C3 pathway.